Welcome to RAAONLINE. Today we're going to discuss about the neuromuscular physiology and pharmacology. Neuromuscular blocking drugs are using regular in the OT, and you must be acquainted with the pharmacology of the neuromuscular blocking agents. Second thing is that this neuromuscular physiology is somehow important. We have to understand different concepts like what is the nature of acetylcholine receptors, how is the sodium channel behaves, what are the two types of gates in the sodium channel, what is the mechanism of action of succinylcholine, how the non-depleting muscle relaxation will act. So this type of things will cover in neuromuscular physiology and pharmacology. Let us start. So here is the diagram. This is a question in the NB, 5 marks question. NBE, neuromuscular junction. Draw a level diagram of the neuromuscular junction is a question. You can see here the motor nerve originate in the ventral horn of the spinal cord or the brainstem. This is the motor nerve. It comes to the muscle junction as the nerve approaches its muscle fibers. Before attaching itself to the surface of the muscle fibers, the nerve divides into the branches that innervates many individual muscle fibers. So here you can see different branches of the nerves and it is attached to the muscle fibers. See, each muscle receives only one synapse. Each muscle receives only one synapse. The motor nerve loses its myelin sheaths and further subdivides into presynaptic boutons to terminate in the surface of the muscle fiber. Coming to the D, the diagram proper, the nerve terminals covered by the Schwann cells. Here the nerve terminals covered by the Schwann cells has the vesicle clusters about the membrane thickness, which are active zones is the membrane vesicles towards its synaptic sites and the mitochondria microtubules towards its other sides. So here is this mitochondria microtubules. A synaptic glutter or synaptic cleft is present in the primary and the secondary cleft separate the nerve fiber from the muscle. So these are the primary cleft, these are the secondary cleft. This separates the muscles fibers from the nerve endings. Okay, so here you can see different things. This is the myelin sheath. This myelin sheath is somehow disappear and the Schwann cell comes into actions. This is the nerve. This is the nerve terminals. There are different mitochondria microtubules are there inside the vesicles or inside the nerve terminals. Here in the synaptic cleft, here you find the enzyme known as cholinesterase enzyme, which degrades the action of acetylcholine. Here we can get different primary clefts and secondary cleft. In the secondary clefts, we have sodium channels. These sodium channels, once activated, they pass the electrical neurons to the muscles fiber and the actin-myosin complex will be stimulated. So these are the active zones through which the acetylcholine will be secreted and the acetylcholine come in the picture. Here in the muscle endings or the muscle, in the muscle endings, we get different acetylcholine receptors. So when we are drawing a level diagram of the neuromuscular junction, you must keep in mind you have to cover everything. So here it is a nerve terminal. In the nerve terminal, you have to write the different mitochondria, microtubules, and along with the synaptic vesicles, you have to draw a synaptic cleft. In the synaptic cleft, to do different primary cleft and secondary cleft. In the primary cleft and secondary cleft, you have to mention the acetylcholine receptors. In the cleft itself, you have to mention the acetylcholinesterase enzyme. Active zone and release site. It is the release site through which the synaptic vesicles will break down and they will release acetylcholine. And once the acetylcholine muscles and the acetylcholine molecule come in the picture, they will enter into the muscle, they will act into the acetylcholine receptor, and finally the sodium channel will be stimulated and they cause the muscle contractions. So it is all about the neuromuscular junctions. So when a brief diagram or the level diagram of the neuromuscular junction is asked in the NB, you can directly put this picture. It is properly elaborated, properly given in the Millers also. You can copy the table or you can copy the picture from the Miller. Otherwise, here it is also there and it is fine. Coming to the systemic illustration depicts acetylcholine receptor. So let us discuss something about acetylcholine receptors. We have three types of acetylcholine receptor. One is immature one, another one is matured one, and some are neuronal in the muscles. So this alpha 7 acetylcholine receptor are also invented right now. Here you can see in the immature channels and the mature channels, the single difference is that here the gamma subunit is replaced by the epsilon subunit. Otherwise, we have two alpha, one is beta, another one is delta. Here also two alpha, beta 1 and delta. Only this gamma is replaced by epsilon. There is a single difference between immature and mature. Otherwise, in the alpha 7 acetylcholine receptor, all the receptors are alpha 7. So mostly we deal with these two type of receptors. And why this immature receptor is very much important for us, because they release potassium ion for a longer time, and whenever we are inducing this patient who are having immature type of acetylcholine receptor, thus there will be the chances of hyperkalemia. Why? Because see, this is the closed state, this is the open state, this is the closed state, this is the open state. Let us differentiate between immature and mature. When this gamma subunit will be replaced by epsilon 1, what are the changes in the acetylcholine receptor takes place? It is low conductance channel, it is a high conductance channel. Second thing is that it is open for a more time. That is, the opening time is very much more. See, it is open for a fraction of seconds or open for a specific time, but here it is open for a larger time. When they are open for a larger time, their conductance of the towards the potassium is very much increased and the patient will develop hyperkalemia. when we induce a immature, and when you are inducing the patient who have large number of immature acetylcholine receptor, there will be very high chances of hyperkalemia. So who are the patients? Mostly the pediatric patients are having immature channels. Mostly the patient who are having Guillain-Barré syndrome, paraplegia, polio type of patients. In the patient, immature channels are very much higher. Second thing, the patient who are the victims of burns, or the burn area is more than 50 to 60%. Then also in the site of the burn, there is the highly densely immature channels are present. So oxide choline to this patient very carefully, because there will be high conductance of the potassium channel and they will release so much of potassium, so there will be chances of hyperkalemia in the immature channels. Okay, this is the low conductance channel, this is the high conductance channels. So they will definitely go and ask what is the difference between mature and immature. Immature having gamma subunit and mature having epsilon subunit.